So now what we're going to do again is talk about what happens to this glucose once it enters in to the skeletal muscle cells from the bloodstream. So we're going to say that this is just after a meal. Okay? Insulin levels have risen due to an increase in blood sugar. Pancreatic beta cells released insulin. Insulin bound to the insulin receptor, triggered the pathway that allowed for GLUT4 translocation, and that glucose in the bloodstream is coming in to the skeletal muscle cell. Again, this is after a meal. I'm not now talking about sort of contraction stimulated glucose uptake. I'm talking about the glucose that comes into our muscle cells after or during a meal. The first thing that's going to happen to that glucose is it's going to get phosphorylated. And specifically, it's going to get phosphorylated by an enzyme that's known as hexokinase. As soon as glucose is within the skeletal muscle cell, this hexokinase enzyme is going to work to phosphorylate glucose. How does it do that? Well, ATP is actually used. A phosphate is broken off, so ATP is hydrolyzed, ATP hydrolysis, and that phosphate is actually added to the glucose molecule, and it becomes glucose 6-phosphate. So what's the purpose of hexokinase essentially adding a phosphate to the glucose molecule that's coming into the skeletal muscle cell? And the answer to that is to actually mostly, there are other important parts to this too, but mostly it's to trap the glucose molecule inside of the skeletal muscle cell. Once glucose has been phosphorylated into glucose 6-phosphate, thanks to the enzyme hexokinase adding a phosphate to glucose, this glucose 6-phosphate can never leave the skeletal muscle cell again. There is no enzyme to reverse this reaction in skeletal muscle cells. There is in liver cells. The liver can release glucose back into the bloodstream. This reaction is reversible in liver cells thanks to the enzyme that exists. That enzyme is actually glucose 6-phosphatase. It removes the phosphate from glucose 6-phosphate and turns it back into glucose so that it can be released back into the bloodstream. But we don't find that in skeletal muscle cells. And it's unique because in skeletal muscle cells, once glucose is within the skeletal muscle cells, it is now incapable of ever leaving again. All right, so now what happens to this glucose 6-phosphate now that it is within the skeletal muscle cell? Well, here you can see glucose, and here you can see hexokinase turning glucose in to glucose 6-phosphate. Okay. After a meal, I'm going to elaborate more on this in a moment, but after and during a meal, largely thanks to insulin, what's going to occur is that glucose 6-phosphate is actually going to get turned into, or added to, I should say, the glycogen that exists inside of the skeletal muscle cell. This glucose 6-phosphate is going to get added to the glycogen chain inside of the skeletal muscle cells. There's many glycogen chains. They're called glycogen granules inside of skeletal muscle cells. But essentially, this glucose is going to get stored inside of the skeletal muscle cell by being added to this glycogen chain. So the question is, of course, how does glucose 6-phosphate actually get added to this glycogen chain? And then the question becomes, why what's stimulating that glucose 6-phosphate to actually get added to this chain after the meal? These next couple reactions I would not ask you about on the exam, but I'm going to say them for clarity. Okay, so here's glucose 6-phosphate. What we have is this enzyme, which is phosphoglucomutase, and what it does is it just scrambles the glucose 6-phosphate molecule a little bit. It changes the orientation or location of the phosphate, and glucose 6-phosphate becomes glucose 1-phosphate. Next, we have UDP glucose phosphorylase. What does UDP glucose phosphorylase do to glucose 1-phosphate? What it does is it adds, you can say this both ways, it adds a uridine or a uracil diphosphate to the glucose molecule, which is this. Okay? Uridine diphosphate is added to the glucose molecule by UDP glucose phosphorylase, and what you end up with here is, you can say this either way, it is either uridine diphosphate glucose or uracil diphosphate glucose. Neither one of these reactions, again, do I care much about for the sake of the exam. Okay, but what's the last thing that we need to do? We need to take uridine diphosphate glucose and add it to the glycogen chain inside of the skeletal muscle cell, and that is done by glycogen synthase. So how does glycogen synthase take uridine diphosphate glucose and add uridine diphosphate glucose, also called UDP glucose, to the glycogen chain? What glycogen synthase does is it takes uridine diphosphate glucose, UDP glucose, and it strips off the uridine diphosphate, okay? Or uracil diphosphate, whatever you want to call it. And what are you left with if glycogen synthase strips off uridine diphosphate? You're left with glucose, right? And then what can be done with the glucose is it can be added to the glycogen chain. And what is a glycogen chain? It's a polysaccharide. It's just a bunch of glucose molecules repeating over and over and over again. It's the storage form of glucose inside of the skeletal muscle cells, or any cell that stores glucose in your body. But we're talking about skeletal muscle cells here, okay? So this is the last step in line, is glycogen synthase. Glycogen synthase is going to actually take the glucose molecule and add the glucose molecule to the glycogen chain by stripping off uridine diphosphate from UDP glucose. Okay? So really what we're interested in is what controls the activation of glycogen synthase. What makes this enzyme very active? Because if this enzyme is very active, ultimately, that is what's going to trigger most of that glucose that's coming into the skeletal muscle cell to be stored as glycogen and not used in glycolysis, the breakdown of glucose to make ATP. Okay? So what we want to talk about is what's the pathway that's regulating glycogen synthase so that it can so that it is very active and making sure that that glucose that's coming in from the bloodstream during or after your meal is actually being stored as glycogen. So a couple things. For glycogen synthase to be active, you actually have to dephosphorylate it. Right? You have to actually strip the phosphate off of glycogen synthase to make it active. A little bit different than some of the things that we've discussed, and this goes both ways. Sometimes you phosphorylate something and make it active, sometimes you dephosphorylate it and make it active, and the reverse can be true for inactivating it as well. And what I'm telling you here is if we want glycogen synthase to be active, we need it to be dephosphorylated. And what's going to do that for us is what's called protein phosphatase. Anytime you see the word phosphatase, it's going to be something that removes phosphates. So we need to activate protein phosphatase in order to dephosphorylate and thus activate glycogen synthase so that it starts storing the glucose that's coming into the cell as glycogen, adding the glucose onto the glycogen chain. The exact pathway by which insulin binding to the insulin receptor activates protein phosphatase, I would not say is all that clear. But what we do know for sure is that when insulin binds to the insulin receptor on skeletal muscle cells, we get an increase in the activity of protein phosphatase. And as you can see over here, what protein phosphatase does is it goes over to glycogen synthase and dephosphorylates it, removes the phosphate from it, and makes glycogen synthase active, which increases glycogen synthesis. If we make glycogen synthase, the enzyme, very active by covalently modifying it, removing a phosphate from it, then the likeliness that those glucose molecules coming into the skeletal muscle cell are going to be stored as glycogen substantially increases because the activity of that enzyme has increased. This is the simpler part of the equation, though. Okay. And again, it's partly complicated because the pathway by which insulin increases protein phosphatase is not all that clear compared to some of the other things that we know about the insulin pathway. But just simply insulin binding to the insulin receptor and making protein phosphatase more active and thus activating glycogen synthase, that's relatively simple. It's this next pathway that complicates things more, but is essential to understand if you really want to understand how insulin does its job with regard to activating the synthesis of glycogen. And that brings us to the conversation about GSK3, glycogen synthase kinase 3. Big thing here, K stands for kinase. That's going to be an enzyme that phosphorylates things. And look at the name, glycogen synthase kinase 3. What GSK3 does, as long as it's active, is it keeps glycogen synthase phosphorylated and inactive. That says less active, which is true, but really, GSK3, its job is to keep glycogen synthase phosphorylated. GSK3, glycogen synthase kinase. Again, it's a kinase that phosphorylates things. Specifically, it phosphorylates glycogen synthase and keeps it in an inactive state. Okay. GSK3 is active when it's not phosphorylated. Okay. So this gets real complicated, and it's just some of this just unfortunately does become just purely a memorization game. But for now, I'll go through this in full detail in a moment. But for now, you have to realize GSK3, when it is not phosphorylated, is active. And what it's doing, GSK3, when it's active, not phosphorylated, is phosphorylating glycogen synthase and thus making glycogen synthase inactive. All right, so why would that be problematic? All right, if insulin is binding to the insulin receptor, and we'll just call it PP, protein phosphatase. If insulin binding to the insulin receptor is activating protein phosphatase, you can see protein phosphatase over here. The job of protein phosphatase is to dephosphorylate glycogen synthase to make glycogen synthase active. But that, again, is problematic because these two enzymes are going to be completely competing against each other. Insulin is going to be binding to the insulin receptor, and protein phosphatase is going to become active one way or another. And let me erase this because it's just getting a little messy. Okay, protein phosphatase is becoming active thanks to insulin binding to the insulin receptor, and protein phosphatase is trying to dephosphorylate and activate glycogen synthase. But GSK3 is still active, glycogen synthase kinase, and it's actively phosphorylating glycogen synthase and deactivating it. So protein phosphatase and glycogen synthase kinase are trying to do the opposite things to glycogen synthase. Protein phosphatase is trying to activate glycogen synthase by dephosphorylating it, and glycogen synthase kinase is trying to deactivate glycogen synthase by phosphorylating it. So here's what we really need to do. We need to knock out GSK3 if we really want glycogen synthase to become active. We want to knock out the enzyme that's trying to phosphorylate and inhibit the activity of glycogen synthase. If we really want glycogen synthase to become active when insulin binds to the insulin receptor so that the glucose that's coming in can be stored as glycogen. So how do we do that exactly? Here's insulin binding to the insulin receptor. I'm not going to go through the whole pathway again, but you know downstream you get activation of AKT or protein kinase B. What AKT or protein kinase B does is, yes, it phosphorylates AS160, but what AKT or protein kinase B also does is it phosphorylates GSK3 and knocks GSK3 out. And if you knock GSK3 out, right, make it inactive by phosphorylating it, what are you going to make easier? It's going to make it much easier for glycogen synthase to become active. Why? Because what also does the insulin receptor activation result in? It results in an increase in the activity of protein phosphatase. And what does protein phosphatase do? It goes over to glycogen synthase and it dephosphorylates the glycogen synthase to make glycogen synthase dephosphorylated and thus active. Okay, so let's put this all together in sort of a nice one, a singular nice discussion is what I'm trying to say. So let's talk about this as a whole, put this all together. Insulin binds to the insulin receptor. If we follow the pathway downstream, we know that we get activation of AKT or protein kinase B. What does AKT or protein kinase B really do? It does two important things for our interest. It phosphorylates AS160, right? Adds a phosphate to it. And by adding a phosphate to AS160, it knocks AS160 out and allows Rab to become active so that GLUT4 translocation can occur. And these GLUT4 proteins can move from the intracellular vesicles to the plasma membrane to allow glucose to come in. Okay? You know all of this. The insulin pathway allows for the uptake of glucose by the skeletal muscle cells from the bloodstream. All right, so what else does AKT or protein kinase B also do? Besides just phosphorylating AS160 to allow for GLUT4 translocation, it also phosphorylates glycogen synthase kinase. Glycogen synthase kinase, what it's constantly doing is phosphorylating glycogen synthase and keeping it inactive. We don't want glycogen synthase to be phosphorylated and inactive. So what protein kinase B or AKT can do is it can phosphorylate GSK3, glycogen synthase kinase, the thing that's keeping glycogen synthase phosphorylated and inactive. And by phosphorylating GSK3, it makes GSK3 inactive, right? It knocks it out. What does that mean exactly? Well, by phosphorylating GSK3, meaning AKT, phosphorylating GSK3 and making it inactive, it's now much easier for the cell to be able to dephosphorylate glycogen synthase and make glycogen synthase active. So we have to finish this off. What also does binding of insulin to the insulin receptor do? It results in an increase in the amount of protein phosphatase inside of the cell, in this case inside of the skeletal muscle cell. If protein phosphatase activity is increased, what does protein phosphatase do? It dephosphorylates protein phosphatase, dephosphorylates glycogen synthase, putting glycogen synthase into its active form. So to summarize, insulin is going to allow for the uptake of glucose from the bloodstream. AKT, or protein kinase B, is going to phosphorylate AS160, knock it out, make Rab active so that GLUT4 translocation occurs. AKT, or protein kinase B, is also going to phosphorylate and knock out GSK3, the enzyme that's keeping glycogen synthase phosphorylated and inactive. The insulin receptor is also going to increase the activity of protein phosphatase, which dephosphorylates glycogen synthase and makes it active. So let me say this. By phosphorylating and thus knocking out GSK3, what the insulin receptor pathway has done is it's made it much easier for protein phosphatase to be able to dephosphorylate glycogen synthase and make glycogen synthase active. Okay. If you don't phosphorylate and knock out GSK3, yeah, protein phosphatase can dephosphorylate glycogen synthase, but then at the same time, GSK3 is going to be phosphorylating glycogen synthase and making it inactive. So the two things are just going to be, these two enzymes are just going to be fighting each other. Protein phosphatase and glycogen synthase kinase 3, GSK3, are just going to be in a battle with one another. So what the insulin pathway can do is knock out GSK3 by phosphorylating it and make it much easier on protein phosphatase to be able to do its job and increase glycogen synthase activity. Okay, so if we increase the glycogen synthase activity, what does that mean? It means that the glucose that's coming in, thanks to the activity of glycogen synthase, that glucose is going to get added to glycogen, right? It's going to increase the activity of what we call glycogen synthesis. Those glucose molecules that are coming in, thanks to the increased activity of glycogen synthase, are all going to get added to the glycogen chain, and the muscle cell is going to store all of this glycogen to use later on. So hopefully this allows you to see that insulin binding to the insulin receptor on skeletal muscle cells is, far more sophisticated even than just allowing for glucose to be uptaken from the blood. Insulin binding to the insulin receptor also allows for the real activation of the enzyme glycogen synthase so that glucose that's coming in can be stored as glycogen. So you know that when insulin binds to the cell, it increases the activity of protein phosphatase, right? These two sides are showing the exact same thing. Insulin binding to the cell, insulin binding to the insulin receptor increases the activity of protein phosphatase. You know that protein phosphatase dephosphorylates glycogen synthase and makes glycogen synthase active. But what I haven't shown you yet is that protein phosphatase will also dephosphorylate this enzyme, which is known as glycogen phosphorylase. Okay, protein phosphatase, it's a phosphatase, so it dephosphorylates things. Not only does it dephosphorylate glycogen synthase, but it also dephosphorylates glycogen phosphorylase. And when you dephosphorylate glycogen phosphorylase, what happens is you make glycogen phosphorylase less active. When glycogen phosphorylase is dephosphorylated, glycogen phosphorylase is knocked out. And what does that mean? Here you can see this in a different way. Insulin binding to the insulin receptor increases the activity of protein phosphatase. What do you know about protein phosphatase? Well, it goes over and it dephosphorylates glycogen synthase, making it active so that the glucose coming in can be stored as glycogen. And what protein phosphatase will also do is it will dephosphorylate what's known as glycogen phosphorylase, and it will then inactivate it. So when you dephosphorylate glycogen phosphorylase, it makes it inactive, the complete opposite of glycogen synthase. Why would we want protein phosphatase to dephosphorylate glycogen phosphorylase and make it less active? Why, when insulin binds to the insulin receptor, would we want to knock out this enzyme, glycogen phosphorylase? That's a relatively easy question to answer because what glycogen phosphorylase does is it breaks glycogen down. What glycogen phosphorylase does is it strips glucose molecules off of glycogen to recreate glucose 1-phosphate so that it can go back to being glucose 6-phosphate and used in glycolysis. Would you want glycogen phosphorylase, the enzyme that breaks glycogen down, to be active when insulin is elevated in the bloodstream and we're trying to store glucose that's coming in as glycogen? Of course not. When insulin binds to the insulin receptor on skeletal muscle cells, its job is to allow for glucose to come in. Okay. When insulin binds to the insulin receptor on a skeletal muscle cell, its job is to allow for glucose to come in and for the glucose to be stored as glycogen. Okay. You wouldn't want that glycogen to be actively being broken down into glucose when insulin is binding to the receptor and trying to allow glucose to come in, to allow for glucose to come in from the blood and be stored as glycogen. So what protein phosphatase does, activated by the insulin pathway, is it actually comes over and dephosphorylates glycogen phosphorylase, making glycogen phosphorylase less active. Right? Protein phosphatase dephosphorylates glycogen phosphorylase, making it less active, and that ensures that glycogen is not being broken down into glucose. And what you know about protein phosphatase also is that it dephosphorylates glycogen synthase and makes glycogen synthase active, so that the glucose that's coming in will be stored as glycogen. So this can be confusing because you're trying to remember so many different things at once and all these different enzymes that all kind of sound very similar. But in the simplest terms, this is really actually easy to make sense of, even if remembering all the different enzymes and what's phosphorylated and not phosphorylated can be challenging. What is insulin doing? Insulin is trying to make sure that the muscle cells can uptake glucose and then store the glucose as glycogen. If it wants to do that, then it needs, well, one, GLUT4 translocation to occur, and it needs the glycogen synthase enzyme to be active. Additionally, insulin isn't going to want the glycogen inside of the skeletal muscle cell to be broken down when it's trying to store the glucose that's coming in as glycogen. So what does the insulin pathway also do? It knocks out glycogen phosphorylase, the enzyme that allows for glycogen to be broken down. right? And again, this, when you say this in the simplest terms, it's easy and it makes complete sense. It's memorizing all the different phosphorylation reactions, dephosphorylation reactions, and enzyme names. That's the challenging part. So what we have established up to this point is that you eat carbohydrates and blood sugar levels rise. That causes your pancreatic beta cells to release insulin. Insulin levels rise in the blood with elevated levels of blood sugar. Insulin binds to insulin receptors on skeletal muscle cells, and that allows, through GLUT4 translocation, for the skeletal muscle cells to uptake the glucose. Additionally, the insulin receptor makes glycogen synthase active so that the glucose that's coming in can be stored as glycogen. It also knocks out glycogen phosphorylase so that glycogen isn't broken down. Okay, so you eat, and the carbohydrates, the sugars that you eat, they end up, some of them, inside of your skeletal muscle cells, stored as glycogen, largely thanks to insulin. And now we transition to the conversation of exercise. Okay. During exercise, muscle contractions, we're going to want to consume the glycogen inside of the skeletal muscle cells. Okay. We want to break that glycogen back down into glucose so that the glucose can be used in glycolysis, the pathway of breaking down glucose to make ATP. So during exercise, what are we going to need to do? We're going to need to activate the enzyme glycogen phosphorylase to break glycogen down, and we're also going to want to inhibit the enzyme glycogen synthase so that glucose isn't being stored as glycogen. Okay, so that's where this conversation is going. It's going to transition into talking about how during exercise, by that I mean skeletal muscle fiber contractions, how skeletal muscle fiber contractions are going to activate the enzyme glycogen phosphorylase so that glycogen that we have due to those carbohydrates that we ate some point earlier on in the day can be broken back down into glucose so that it can be used in glycolysis to make ATP and power those contractions that are occurring inside of the skeletal muscle cells. Because those skeletal muscle cells are contracting and consuming more ATP than they normally do, right? So now they need to make more ATP to meet their ATP demands. And depending upon the muscle fiber type, that can happen in large part through the breakdown of glycogen to glucose into glycolysis to make ATP.